Hi, my name is Kateryna Voitiuk. I'm a bioinformatics PhD student at the University of California, Santa Cruz, and I would like to share my work with you about a hardware and software platform that I created to optically stimulate neurons. The human brain has billions of neurons. Each neuron has connections with other neurons via synapses, and together they form vast networks of communication. There are many different kinds of neurons, but they all have a similar architecture. Neurons can be stimulated by other neurons, but we can also perturb them by electrically zapping them or genetically engineering them to be stimulated by light. Using this principle, we built an optical stimulation platform using a commercial high-density microelectrode array and off-the-shelf electronic components. We also 3D printed an insert to guide an optical fiber over the neurons. We wrote code to deliver the stimulation at precisely aligned intervals, aligned to the data, and created functions for configurable stimulation protocols. Next, we did an experiment where we programmed stem cells to grow into a little piece of cortex called a cortical organoid, and infected this cortical organoid with an optogenetic gene that after 3 to 7 days became expressed in a subset of excitatory neurons in the tissue. We stimulated the neurons over time with increasing light intensity. And here we see a 2D dense grid of electrodes with the neurons sitting on it and zoomed in here. And the colors represent different neurons that are intertwined on top of each other. The neurons show spontaneous activity in the beginning. And then when the light stimulation happens, some neurons highlighted in green are later firing on the onset of the light. In the red graph, we also see the firing rate of all the neurons peaking during the light stimulation periods. Here's one neuron in a different experiment that has this characteristic waveform in blue. And under constant intensity and frequency, we saw that this neuron is getting tired over time, as expected. We're also using this optogenetic platform with collaborators at UC San Francisco to deliver optical stimulation to brain tissue samples from epilepsy patients to understand how we can treat seizures with light. Thank you so much for ARCS for sponsoring this work, and I look forward to talking to you at the poster session.